So now I want to go a little bit into the detail. Now we spoke about the very big frame of experiments, and we need to go a little bit more in detail about the experiments. And we have to talk about how an experiment is done. What are the common elements of all experiments and how they can be, and what are the possibilities, what needs to be included in an experiment, and what needs to be excluded. So the first characteristic of an experiment is one, that is the hypothesis. So we need to always start from an hypothesis. The hypothesis is related to the aims. So we define an hypothesis, and then according to that hypothesis, we define the aims. So the hypothesis is the question we want to ask to the data. It must be relevant, it must be clearly defined, it must be specific, mainly it must be measurable. It must be realistic and achievable. We cannot ask the data something which they can't answer. And it must also be organized in a timely succession. I mean, we need to have clear what we want to achieve first and what we want to achieve later on. Sometimes there are some results which can only be achieved after some preliminary work, and we need to have this clear in mind when we make an experiment. I see a lot of experiments where we can see that the initial hypothesis was not clear, and you see that in such experiments the questions were asked at runtime, let me say runtime. So we see that the authors were going on, and while they were going on, they were asking questions. And it is much more elegant when we see that all the questions were really clear in advance before making the experiment. That is an elegant experiment. In the other case, we have the idea that the authors were somewhat blind. They went on without having a clear idea where they wanted to go to. And this is not really elegant. So questions needs to be clear in advance. When the questions are clear, we allocate, we impose, we decide the experimental stimuli. So we need a definition here. What is an experimental stimuli? Experimental stimuli are usually known as the experimental factors. So this is the way we talk about the stimuli, experimental factors, or simply factors. Factors are the treatments. When we have a comparative experiment, one factor has more than one level, and the levels are called experimental treatments. So one factor might be the genotype. So we want to compare different genotypes. This would be the factor. Then we have the levels. So within the factor genotype, we have the level A, the level B, the level C, the level D, and so on. So we have 1 factors and 4 levels. These are called the experimental treatments. We have 4 treatments for 1 factor, right? So you need to get used, if you are not already, you need to get used to this kind of namings, because I will use them from now on I will talk about experimental factors, and I will talk about experimental treatments. Sometimes we have more than one. We might have 2 factors. In this case, we talk about factorial experiments. So factorial experiments are those experiments where we have more than one factor. For example, think about an experiment where we want to compare genotypes and fertilizations, right? So we have 2 factors. Now we need to combine the 2 factors, and we can combine them in 2 ways, 2 different ways. We can have crossed factorial and nested factorials. When we have crossed factorials, we include all the possible combination of factor levels. When we have nested factorials, we do not include all possible combinations. But I will give you an example, otherwise I assume that this is not clear. This is a crossed factorial. We have an experiment with 2 factors, the genotype with 3 levels, A, B, and C. So this is still Italian here. And we have fertilization type, 2 levels. For those who are experienced, these are 2 different fertilizers, urea and poultry manure. For those who are not experienced, it doesn't matter. There are 2 different fertilizers. So we have 2 factors, and we can combine the levels, and we can include in the experiment 6 possible combinations. Genotype A with poultry manure, genotype A with urea, genotype B with poultry manure, genotype B with urea, genotype C with poultry manure, and genotype C with urea. So you see 6 combination levels. In this case, the design is crossed, right? Then we can have a nested factorial. This is an example. In this case, I want to compare 2 plant species with 2 levels. I want to compare barley and wheat. I'm not sure for whether you know these crops. These are 2 cereals. So they are different crops, and they have different genotypes. So if we want to compare the genotypes, and we have 3 levels, the levels must be different for these 2 species. For barley, we have the genotype A, the genotype B, and the genotype C. And these are for wheat, we have the genotype D, the genotype E, and the genotype F. Here again, we have 6 possible combinations, but the second factor levels are different according to the crop. So we have barley A, barley B, barley C against wheat D, wheat E, and wheat F. So the design is nested. It cannot be crossed because certain combinations are clearly missing. So it is rather important to be able to recognize a nested experiment from a crossed experiment because the data analysis is different. And we will see this maybe in a few weeks. So one of the levels can be the so-called control or check. The check is usually included as the reference for all the other treatments. We might have different types of control. The control might be the untreated level, if we have it. Or we could use a control treated with a placebo. This is not really very often done in agriculture. It is mainly done in medicine. Then we treat with a placebo because we want to avoid some psychological effects relating to the fact that one is assuming a particular drug. In agriculture, very often the control is treated according to the usual practices. For example, if we want to compare wheat genotypes, we usually include a reference genotype, which is the one that is normally grown in that location where we make the experiments, right? So this is what concerns the experimental treatments. But then we have to talk about the subjects. Once we have decided which treatment we want to compare, then we need to select the subjects which or who, if they are humans, are going to receive the treatments. So our definition is this one: the smallest units receiving the experimental treatment independently from all other units. Let's recall the pseudo-replicates. These are not true replicates because they do not receive the treatment independently from one another. So this is what defines the experimental unit. What is important, we know that already, the experimental units are sampled from a wider population of units. We almost always sample. We almost never include all potential subjects into an experiment. Well, in some cases, yes. For example, I have friends from the technique department. Sometimes they have just a few units to allocate the treatment to, so they simply use all units they have. This is why I'm working with very rare breeds or whatever. In that case, they have a few units and they use all units they have. But that's really an exception. Normally, we sample subjects from a wider population. Once we have defined the treatments and the experimental subjects, we need to define the response variables. Well, this is something I forgot to translate. This would be what we record. We can record classes. This is it is not so common. I mean, in some cases we measure traits, and when we measure, we talk about biometrical assessment. We measure. What do we measure? We measure height, yields, the level of sugar in blood, whatever. But we measure. In some other cases, we made a sort of a classification process. So we take the subject, we do not measure anything, we simply assign them to a certain class. Just think about sex, for example, or think about the color, or think about other classes when we make treatments against weeds, for example, when we kill plants, we can record whether they are dead or alive. With seeds, we can record whether they are germinated or not. So we have just classes. We do not measure. And this is still possible. In some cases, we made sensory assessments. I mean, we go look at a subject and grade it or him into categories, or we express our liking. That would be the Likert scale. Do you like this cake? And you might answer yes, so and so, or no. So you made 3 classes, and you make these 3 classes by using your eyes or smelling or whatever. Well, this is very common, for example, for wine testing or for other types of testing. I remember a few years ago, we were trying a product for chemical controlling tobacco, the lateral ears of tobacco. And these products usually smell very much. And a firm was trying to find some alternative compounds which did not smell that much. So what we made was just sensory assessment. We went and in some way classified these products based on their smell. So we used our sensorial system, right? This is still possible, and it is very, very common in some cases. In some cases, for example, we evaluate weak controllability or the efficacy of some drugs simply by looking at the subject. We don't need to measure. We just look at the subject and decide how it should be classified. So that's very common. It doesn't cost much, actually, and it's very quick. So depending on what type of assessment we do, we can have different types of variables, of response variables. And it's important to be able to recognize them, because depending on the type of variable, we select the method of data analysis. We can have nominal or categoric variables. In this case, we have classes. And we have only one trait, one characteristic of these classes. They are mutually exclusive. Nothing else. There is no order. There is no distance between classes. We just know that one subject either is in one class or it is in the other one, but it cannot be on both classes because they are mutually exclusive. This is an important trait, nothing else. These variables are rather common. Then we have ordinal variables. In this case, we have classes. They are mutually exclusive, and they are also ordinal. I mean, they can be put in order from the lowest one to the highest one. What is an example of an ordinal class? In some case, a few years ago, I worked with the botanist. They were, in some cases, looking at the years of a particular plant, and they were looking at the number of caryopsis in one flower. They could be 0, 1, 2, 3, or 4. These are ordinal classes. These are mainly counts. We will see them in a few, but these are ordinal classes. We can also have other ordinal classes. For example, if we want to score the taste of a wine, we can use a scale where we have 1 when the wine is not good, 2 when it is better, 3, 4, 5, from 1 to 10. So these are ordinal classes, because we know that if we give a score of 3, that wine is worse than when we give a score of 4. So they are ordered, naturally ordered. They are like counts or ratios, because counts and ratios are exclusive They are ordered, but they might have one further trait, that is the distance. If we have classes made with counts, like in the example I did before, we have 1, we might have 2. We know that 1 is lower than 2, but we also know that the distance between one class and the next one is constant, because we have the same distance from 1 and 2 and the same distance from 2 and 3. And we know that the distance from 1 to 3 is twice as much the distance from 1 to 2. This is another important property because it allows us to make some calculations. For example, we can calculate the mean or the average or the deviance. We cannot do so with nominal categories. So it's very important. With quantitative variables, which is the last type, we have another important characteristic apart from the exclusivity, the order, and the distances. We have the so-called continuity. We have no classes. We have only one continuous variable, which might, at least in theory, take any value from a minimum value to a maximum value. Right? So these are the types of variables you can meet in your work, and you need to be able to recognize them because they are analyzed differently. So just a few words about sensory and visual assessment, which are very common. They have advantages and disadvantages. When I began doing this work, I was rather skeptical about sensory and visual assessment because I was thinking, how can we be precise if we evaluate our subject simply by visual assessments? In reality, I have learned that if with some training, we can give rather precise assessments, even visually. We need experience, of course. And if we have experience, these assessments have strong advantages because they have low costs, high speed, and they do not need instruments. These are the assessments which are favored by industries, for example, because they give precise information without the need for high costs. Of course, they have disadvantages. Sometimes they are not precise. Sometimes they might be subjective. I can give a different assessment by another, a difference from that's given by another person. I can be unconsciously influenced by knowing the experimental treatment. That happens. I have to score field plot, which was treated by a herbicide, which I know. I think I know how it should behave. And when I go and score the plot, I am influenced by the fact that I know how it should behave. Furthermore, when we do assessments, we tend to get tired. And when we get tired, we tend to lose our uniform judgment parameter. So it might be that when the time progresses, we lose our efficiency. And of course, we need a specific training. While I can take everyone and tell him, go and take the measure of that plant, because he only has to measure a plant. Everybody knows how to do this. I cannot tell everybody, go and score that plot and try to assess the width controllability, because that's not easy. You need to be trained to do that. So I'd like to talk about some peculiar aspects of field experiments. But perhaps before I do this, we may take 10 minutes break to have a coffee. What about this? First of all, if you have questions, just ask me, and then tell me whether you agree that we make a short coffee break, 10 minutes. Now we're going to talk about a part which is perhaps of little interest for those who are in the psych. All right, so there is one question. I was thinking about the sex of the subject, which kind of variable is. What about you? What is your opinion? What is your opinion about the sex? What type of variable is it? Does it express? Yeah, that's correct. It's nominal or categoric, whatever, because sexes are mutually exclusive. I mean, you cannot put one animal or whatever in both classes, and but there is no ordering, and there are no distances, and there is no continuity. So sex is certainly a nominal variable. Nominal or categoric. We use both names for these classes. Other questions? Well, I was just saying that this part I'm going to talk about now is very interesting. Not very interesting for those of you who are in the psychology field, maybe, but it is interesting, very important for those of you who are in the biotechnology field, especially if they want to become plant breeders. Then they have to make experiments in the field to compare genotypes, for example. And so the first aspect you are going to decide or to select is the field. I mean, you have to select the field. And this is really easy to tell you. It must be easy to reach. It must be continuous. So we need to avoid every possible source of variation. Water stagnation, ditches, rows of trees. So we need to make sure that the field where we want to make the experiment is as homogeneous as possible. And it must have also the appropriate history, because maybe we had a crop in the year before which we left a lot of fertility, and that might not be good. Or we had a crop which left the soil with little fertility, and this is not good as well. So we need to have a soil with a field which is very homogeneous and with the appropriate history. What we do very often to increase field homogeneity is, for example, to use a previous homogenization crop, for example, forage, or for example, oat, some crops which use a lot of nitrogen so that the soil is not that fertile. We used to, for example, make deep tillage in order to put all the seeds of weeds under the soil so that we have no problems with infestation during the experiment. We do not do an experiment twice in the same field. An experiment always creates dishomogeneity, and therefore using the same field twice is not recommended. Sometimes, if necessary, we sow weeds and spread pathogens, if it is strictly necessary. This is something we can do in the agricultural field. Of course, we wouldn't be able to do so in other fields working with humans or animals. Another thing we do very often is to sow at high densities and thin out after the emergence. We do this to make sure that the crop has always the right density. Otherwise, the experiment fails or may fail. It is important that we recognize the difference between two types of experiments in the field. We can have the so-called on-farm experiments. And usually these on-farm experiments are typical of the final stages of development for new techniques. When we have a new technique and when we know pretty well that this technique has advantages, then we need to make on-farm experiments to evaluate this new technique in realistic conditions using the traditional and regular farm machinery. These experiments are very important, but they are usually characterized by high realism but low homogeneity, so low precision and low power. So it is very difficult to prove that a difference exists because we are subjected to all sources of variability which are typical of working in the field. Nonetheless, these experiments are very important because they tell us whether the new genotype, the new tillage method, the new fertilization method is really convenient when used in real farm conditions. We might have found significant effects during laboratory experiments, but then we have to transport these results into the field. And in some cases, what seemed to be convenient in the laboratory is no longer convenient in the field because it increases the costs without giving any real and significant advantage. When we make on-farm experiments, we use the strips. What you see here is a big field, an irregular field, which is typical of certain situations. For example, here in the hills of Umbria, our experimental subject is a field strip. You see, here in red, you have a strip which was treated with A. For example, it was sown with a particular genotype, or it was cultivated with a particular technique. Here we have the strip B. In total, we have 4 treatments, which are labeled with the letters from A to D. You see A, red, B, green, C, blue, and D, that is brown. And you see we have 4 strips, and they are repeated 4 times. So we have replication. So this is a valid experiment. And you see that they are randomized, because in the second replicate, we have a different ordering. We have B, D, A, and C. Then we have the third replicate, and the ordering is again different. D, A, C, B. And then we have the fourth replicate, D, C, B, A. So again, different ordering, because we randomize every time. So this is the layout for a non-farm experiment with 4 treatments by 4 replicates. So we have 16 subjects in total. These strips are rather big. They might be 1 hectare, for example, or even more than 1 hectare. But they are big enough to be able to use the usual farm machinery. So we don't need to use specific experimental machinery. We work as a farmer would work in the same conditions, right? In my case, we work more with small plot experiments. So instead of working with big strips, we use small plots. And therefore, we move in an intermediate position between farm experiments and laboratory experiments. So we are not really in the same far condition, but we are close to it. But nevertheless, we have a small plot, so we can be very accurate in a small plot. In this case, the aim is not to appreciate whether a technique is convenient in farm condition. We want to gain knowledge, but we want to gain such knowledge in the agricultural environment, so in the natural environment. The plots are rather small, so we use specialized machinery. And by using this machinery, we achieve good homogeneity, good precision, and good power. So if we have good power, we can likely discover significant differences. Right? So just to show you, this is a small plot experiment. And this is grain sorghum. I don't know whether you know this crop because it's not a very common one, but the picture is nice because you can see the different genotypes, which you can tell by the different types of spikes. And you see different plots with different genotypes, and you can have a feeling for the size of the plots, which I'm going to talk about now. So this is a small plot experiment. Plot sizes from 10 to 40 square meters. If we have low density crops, we can have smaller plots because we have a lot of plants. For example, think about wheat. Wheat requires 350 plants per square meter. So with 10 square meters, we already have 3,500 plants, which is a lot for an experiment. If we think about maize, for example, maize has a density of roughly 7 plants per square meter. Clearly, if we have 10 square meters, we would have 70 plants, which is not that much. So we have to increase the size of the plot, reaching up to 20 or maybe better, 40 square meters. Well, with fruit trees, we usually use plant rows. We don't really use plots. We use plant rows, plant branches as plots. We use a different arrangement. If we make smaller plots, the plots are more homogeneous, but they can become little representative, which is not good. So this is the selection of plot size is always a trade-off between homogeneity and representativeness. In some cases, we make smaller plots, but we increase the number of replicates. So instead of making 4 replicated plots of 40 square meters, we make 8 replicated plots with 20 square meters. That's a possibility, which we decide at any time. The plots are usually of rectangular shape, and there is one important reason. We need to select a width which is proportional to the width of the machinery we use for sowing, harvesting, spraying. Otherwise, we have to go up and down several times, which is not really handy, and it may give some sorts of imprecision, which is what we wanted to avoid. Therefore, if we have a sowing machine which has 2.5 meters width, we usually have a plot width of 2.5 meters, so that we can sow just with with one passing. So plots are rectangular, and we need to have one plot. The plot is the subject, so we need to have replicates. And with field experiment, we usually have from 3 to 6 replicates. That's the normal number of replicates. Why not less and why not more? It is a trade-off between precision and costs. The highest the number of replicates, the highest the precision, but the highest is the cost. So, and if we have a lot of replicates, the experiment becomes very big and we may not be able to manage it because we have to sow, we have to harvest, we have to spray herbicides, we have to fertilize, and we do this usually by hand in small plots. And if we have, say, 300 plots, then we can't do that in one day unless we are 20 people working, which is not the normal situation with agricultural experiments. We have 4 technicians, for example, in our department, and they have to do all the work. One peculiar aspect of a small plot experiment is the so-called border effect. That's very important. If we take one plot and consider the edges of the plots, the plants along the edges are not in the very same conditions as the plants in the middle of the plot. For example, the plant along the edges may lack competition because they do not have a plant on one side. They do not have crops on one side. Or they can have excess competition because they might have a taller plant on the neighboring plot. So they suffer from excess competition. Or they might have other effects from neighboring plots, e.g., for example, if I spray one fertilizer, this fertilizer might affect the plot which is immediately next to the treated one. Or if I spray an herbicide, this might affect the edge of the next plot. So, border is always a situation very difficult. And we need to avoid border effects if possible. But that is not possible. And therefore, we try to minimize border effects. And how do? We usually use some techniques. For example, we remove plot edges when we harvest the plot. We remove plot edges and only consider the central part of each plot. This is a very important technique. We don't collect the edges. We just collect the central crop rows. We might leave a lace between plots to minimize the neighboring effect. So we leave some space or we remove a lace. It depends on the type of experiment. So either we remove a lace, so we make the crop, the 2 plots, one next to the other, very close, or we make them a little bit more distant and we put the crop in between, so that we avoid as much as possible the border effect. We surround the experiment with untreated plots to avoid effects at the edges of the experiment. Then there are a few items which were not translated, but they don't need to be translated because they need to be erased, but I forgot that. So here we go. When we are done with all this something like developing the protocol for the experiment, when we have decided how many treatments we have, how many replicates we have, and therefore we have decided how many subjects we have, because the number of subjects is always the product between the number of treatments and the number of replicates. When we have decided how big are the plots and so on, we usually draw the so-called field map. This is the field map. This is an experiment where you see we have 32 plots, and they are clearly identified by using numbers. Then we need to specify the size of plots. And you see that these plots are 2 meters wide and 8 meters long. So in total we have 2 by 8, 16 square meters per plot. Here we have put an arrow which shows the northward direction. This is very important because when you go to the field, you need to know where the first plot is located. You see that I have surrounded the experiment with border plots. This is to minimize the border effect along the edges of the experiment. This is what we do regularly. And this is even important for us because we have to give this plan, this map to the technicians who are going to design the experiment. We might be able to go together in the field to see how we can do this. We have to get a bit organized with transport, but we will talk about this in a few weeks. Once we have designed the map, we have to decide to which plot we allocate which treatments. So we have the so-called treatment allocation. Treatment allocation. So which subject is to be treated with each treatment? Now, the allocation of treatments is really fundamental. And if you take the old books about the experimental design, they name this allocation of treatments as experimental design. So something as if the allocation of treatment was the only important item about experimental design. Of course, we know that the experimental design is much more than allocating treatments. But nonetheless, allocating treatment is the central point of experimental design. And we need to carefully select how we allocate the treatment. Just to tell you, this is very important. In some cases, this is not really agriculture, but in some cases, we need to allocate treatments in a sort of, let's say, secret manner. I mean, we can make so-called single-blind experiments. With single-blind experiments, the experimental units are not aware about the treatments. This is very common, for example, with medicine. In some cases, the experimental unit knows that he's going to be treated, but he doesn't actually know whether he's going to receive the treatment or the placebo. Right? That happens. Of course, we make single-blind experiment when the experimental unit can be influenced by knowing which treatment he's going to receive. This is not common with agriculture. With agriculture is more common the other situation where the experimenter doesn't know what treatment has been assigned to a particular unit. We use such a scheme when we spray herbicides and then we make visual assessments. Then it is really very dangerous to know in advance what treatment has been assigned to a certain plot, because we might be influenced when we make the assessment, the visual assessment. Therefore, we do not know, when we go and make the assessment, we do not know about the treatment for a certain plot. If neither the unit nor the experimenter are aware about the treatments, we talk about double-blind experiment. So we might have single-blind experiment and double-blind experiment. Or we can have open experiment where everybody knows everything, which is the common situation in agriculture. But I think it's important to at least give a brief mention about this single-blind and double-blind experiment. So, first type of design. This is very important. I can allocate treatments totally at random. This is what Fisher told us. And this is rather common, but not so common, really. We will see that. In any case, it is very simple. I show you the map. Look at this map. Here, I have allocated 8 treatments with 4 replicates. Look here. For example, A. The treatment A has been assigned to the plot number 3, plot number 19, plot number 29, and plot number, here we go, 32. So we have 4 plots treated alike, 4 replicates. We have 8 treatments, 4 replicates, 32 plots. That is exactly the size of this experiment. Everything is allocated totally at random. This is very simple. This is correct, but every possible difference between plots in terms of fertility or whatever is not recognized, because we have only one known treatment factor. All the rest is experimental error. We call experimental error, differences between plots, original differences between plots. Of course, the plots treated alike, A, will give a different yield. It doesn't matter how precise we are. And we have no way to account for such a difference, because these plots were treated alike. So differences are only due to unknown effects, and therefore our precision will be rather low. But that's not dangerous, because we have seen that precision is important, but accuracy is more important. In this case, we have a low precision, but that doesn't matter as long as the accuracy is okay. But we can see that there might be problems with accuracy too. For example, just imagine that this first column here is less fertile than the second column, that is less fertile than the third column, and so on. Therefore, let's imagine that there is a fertility gradient from left to right. Am I clear? If this is true, we are having some unwanted systematic effect. Look at C. In the case of C, we have 2 replicates on the less fertile part. Look at H. In the case of H, we have only 1 replicate replicate in the less fertile part, and we have 3 replicates, look at here, 3 replicates in the most fertile part. So you see that by assigning treatments at random, we can create systematic error. And if we create systematic error, then we lose accuracy, because we are making advantages to one treatment and we are making a disadvantage to another treatment. So we are confounding the treatment effect with the fertility effect. And this is a very big risk with completely randomized design. So the question is, the fact is that completely randomized design are good, they are valid, but they are okay only for very homogeneous units when we have no knowledge about possible gradients of fertility, of height, or whatever. That there have not to be gradients, because if there are gradients, then we run the risk of making some systematic error. I think this is a very important point, and I'd like to stop for a while to ask you whether you have questions, and please give me some sort of feedback to let me understand that you are there, you are alive, and you are understanding what I'm talking about. No feedback? That's very dangerous. Are you there? Let me write, because so I have 4 feedbacks. I have Claudia, Fabio, and Valeria. Yeah. Someone is coming, everything clear. So you need to make sure you understand the advantages and disadvantages of completely randomized designs. Okay, thank you for your feedback. Thank you now. Good. So I wanted to take the time to have my coffee. Let's go ahead then and make it slightly more. Yes, you can, Hugo. Yes, you can. And I would tell you that you should. You should make all possible interventions to have plots which are as homogeneous as possible. You need to do that. Of course, in some cases you simply can't, because maybe you don't own the field, you're working in a farmer field, and so you can't do much. Then the selection is more important than the intervention. You need to select a uniform field, and if you can, you should perform all intervention to make it as homogeneous as possible. Okay, so what I could introduce to avoid systematic errors is the so-called blocking. That is a very important practice. I want to be clear, it does not necessarily relate to field experiments, but in field experiments, it is very common. But you can use it with all types of experiments, even in psychology. What is a blocking factor? A blocking factor is an innate factor which is different across units, but it is not allocated. So, you see, if we go back to the previous design, we can see that if it is true that the columns on the left are more fertile than the columns on the right, there is an innate factor, which is the fertility, which we did not allocate, but still it makes a difference between plots. That would be a blocking factor. And I can exploit that factors because I can split the units in uniform groups, which I call blocks. So in this case, I could make a first block with 8 units here, and the second block with 8 units here, and the third block here, and the fourth block here. So I could divide the units in blocks. Then what I do, based on what aspect? Well, in that case, I could split the plots based on fertility, but I could use positioning, similarity, sex, height, age, or whatever. I take the units and split them in groups. Each group must contain as many individuals as there are treatments in the experiments. So in this case, we have 8 treatments. We need 8 individuals per block. It is not always so. In some cases, we can adopt incomplete blocks, but I'm not going to talk about these complex experiments. So if we have as many units in a block as there are treatments, it is clear that one block is one replicate, and we have as many blocks as there are replicates in the experiment. I give you an example, then you can ask me questions if that is not clear for you. Here, the same experiment, 32 plots. I divided them in 4 blocks with 8 plots each, and you can recognize the blocks by the colors. In the first block, I have all 8 treatments in randomized order. In the second block, I have all 8 treatments in randomized order, and again, the third and in the fourth block. So I'm repeating the experiment 4 times. What is the advantage? Take the treatment A. I have the treatment A here in the first block, in the second block, in the third block, and in the fourth block. And therefore, the treatment A is put in all different fertility conditions. So in this case, I can avoid any systematic effects because all treatments are in all fertility conditions. But again, it might be age, it might be sex, whatever. I'm putting one treatment in all possible conditions. So I do not run the risk of making an advantage for one treatment and a disadvantage for another one. Is that clear for you? We will see that the analysis of data for randomized experiments, completely randomized experiment and complete randomized blocks designs. Perhaps I should write here, that's again Italian, randomized complete blocks design. So the abbreviation, the correct abbreviation is RCBD. Right? Another advancement, that would be the Latin square design. In this case, we can have two blocking factors. Look at this design. In this case, I had to make it slightly smaller for reasons that I will tell you about in a few seconds. In this case, I only have 16 plots, and I have 4 replicates and 4 treatments, A, B, C, and D. I might have a fertility gradient in both directions, along the columns and along the rows. So I might have more fertile plots to the left and to the top. In this case, a Latin square design is a design where I put every treatment in all rows and all columns. Look at the treatment A. It is put here in the first column, 4th row, here in the second column, 2nd row, here in the 3rd column, 1st row, and here in the 4th column, 3rd row. So you see, it is in all rows and all columns. In this case, I can avoid possible confounding due to the fertility gradients because I'm keeping into account both fertility gradients. Some students usually tell me that this is the Sudoku game. It's like a Sudoku. You mean you have to put all treatments so that they are in all rows and all columns. I put them in different colors just to let you have the feeling for the different positioning of treatments. You see the C, all rows and all columns, and the D, all rows and all columns. Again, you see the size of the plots, the north direction, and the border around the experiment. Why did I have to make this smaller? I had to make this smaller because I need to have as many blocks as there are treatments and as there are replicates. So in this case, I needed 4 replicates and therefore 4 treatments. Otherwise, it is almost impossible to have all treatments in all rows and all columns. That's why this is called a Latin square, because it has a square form. I mean, it's rectangular because the plots are narrow and long, but still it is a square because we have 4 columns and 4 rows and 4 treatments, right? So this is okay. This is very good because we have very high precision and we can avoid as much as possible systematic effects. But on the other side, this is okay only for very small experiments with very few treatments. Otherwise, we need a lot of replicates because we need to have as many replicates as there are treatments. So if I have 8 treatments, like before, I need 8 replicates to design the Latin square. Well, it's important that I show you another example, just to tell you that we can use Latin square designs also outside agriculture. In this case, just assume that we have a certain task that can be accomplished in 4 different ways. And we have to decide which is the quickest protocol. Okay, is that clear? I mean, a certain task might be plowing a field or whatever. It might be a task like writing some sentences or whatever. We have 4 ways to do this task, and we have to decide which is the quickest one. Now, in order to do this experiment, we have 4 different ways. If we want to have different subjects, perhaps we might need, if we want to have 4 replicates, we have 4 treatments, 4 replicates, so we would need 16 subjects. We might not have 16 subjects available. Therefore, we might be interested in making such an experiment with 4 subjects. Right, so we have 4 subjects and 4 treatments. Clearly, we cannot assign a different treatment to each subject, because this would be an unreplicated experiment. What we can do is to have one subject work with all protocols, one after the other, right? So we take one subject and we tell him, okay, now you use in randomized order. You use the protocol A, then the protocol B, then the protocol C, then the protocol D. This is really okay, because if there are differences in proficiency among subjects, we have no advantage, because every subject works with all protocols, right? So this would be a sort of randomized complete block design, because every subject is a block, and within the block we have all 4 treatments, all 4 protocols. Is that right? So here, first possibility, different proficiency for different workers. I can have each worker accomplish the task with all 4 protocols in 4 randomized turns. So in this case, the worker is the blocking factor, because within each worker we have a blocker, and within we have the 4 treatments in randomized order. This is one possibility, but we have one problem. The worker gets tired and the latest turns are penalized with respect to the earliest one. I mean, if I use the protocol A in the first turn, I'm not tired and I would probably be very quick. If I use the same protocol in the last turn, I've been working with all the other 3 protocols before, and I am tired, and probably I will be slower. And in this case, the last protocol is penalized, and we need to avoid that. So what can we do? We can use a Latin square design. So we can have the 4 workers using all protocols in randomized order, but we need to be careful that each protocol is used in all turns. Look at there. We have the protocol A, which is used in the first turn by the worker 1, in the second turn by the worker 2, in the 4th turn by the worker 3, and in the 3rd turn by the worker 4. So we see all treatments in all workers and in all turns. This is a Latin square. Nothing to do with the field, but still a Latin square design. Is that right for you? Is that clear? You have any questions about this design? So we've been talking about 3 types of design, experimental design so far: the completely randomized design, and the Latin square design, and the randomized complete block design. So 3 designs. We can have more, and we will talk about these further designs in a few seconds. But if you have questions, just ask me, as long as I drink some water. Next design I want to speak about is for factorial designs. May I just remind you that factorial designs are those designs where we have more than one factor. We can simply design factorial experiments by using completely randomized designs or complete randomized complete blocks design. Need to make a correction here because this is CBD. But there might be some specific designs for factorial experiments. Now I'll make you an example, which might be clear for those of you who work in the agricultural field. For the other ones, maybe it's not as clear, but I guess it might be understood. If you don't understand it, just stop me, because I'll make another example, or I'll try to find something which is similar. Think that we have to compare 3 tillage methods. Farmers used to till the soil. Maybe you know that. And we have several tillage methods, and we want to compare 3 of them. I used 3 abbreviations. MIN stands for minimum tillage. SP stands for shallow plowing. DP stands for deep plowing, but that doesn't matter. These are 3 tillage methods. Second factor is the weed control method. And in this case, we have 2 abbreviations, TOT and PART. TOT stands for total weed control, broadcast all over the soil surface. PART stands for partial control. We control weeds only along the crop rows. So we have 2 factors. The first one has 3 levels, and the second one has 2 levels. Therefore, we have 6 possible combinations. So we have mean tot, SP tot, DP tot, and then we have mean part, SP part, and DP part. I guess this might be clear. 2 factors. One has 3 levels, the other one has 2 levels. 3 by 2 is 6, so we have 6 possible combinations. And the design is crossed. If you remember what we talked about yesterday. This design is crossed because we have all possible combinations. Am I clear? Is this clear also for psychologists and PhD students in veterinary science? I'm just waiting for a swift feedback from you, because if this is not true, well, I'd rather not go ahead. No feedback? Everything's clear? So at least I have one feedback saying that it is clear. Is clear for all the other ones? Or maybe we have no psychologists today and no, well, another one came. Okay, so that's good. That's good. Well, I'll go then. So look at this design. This is a map. We have 6 combinations, 4 replicates. So we should have 6 by 4. 6 by 4 is 24 plots. And you see they are arranged in 4 columns and 6 rows. And you see that each column is a block. And within the block, there are all the possible combinations. BP part, and they are randomized. SP tot, mean tot, DP tot, mean part, mean tot, and no, well, this is DP. There must be something wrong with the codes, because this cannot be mean tot. There must be some mistake here, because this must be mean, and yes, it can be mean tot. SP, DP, tot, tot, 1, 2, 3, tot, 1, 2, 3 parts, so that's okay. Then we have DP and DP, and this is okay. We have mean, and this is okay. So no mistake, it's okay. We have all 6 combinations in the first block, and there must be all the 6 combinations in the other blocks too, and you can tell this by the colors. So this is a completely, again, it needs to be corrected here. This is a randomized complete block design. And it is a factorial design. What's the problem with this design? We need to work the soil. We need to till the soil. Those of you who have an experience with agriculture, they know that tillage machinery is rather big. And we need to plow this plot here. So we need a lot of space between one plot and the other one, because we need to have space to get the machinery out of the soil without invading the next plot. So we need a lot of space between one plot and the other one. And this is a problem for us, because the experiment becomes bigger and the soil heterogeneity therefore increases. So you see, we need 2 meters width for the plot, 8 meters length, but then we need 4 meters from one plot to the other one, and 3 meters here. So this is a perfect design, but it is not really handy for technicians. And perhaps precision decreases because we have a lot of soil heterogeneity, right? So there are some other ways we could plan such an experiment. I'm going to read this slide here. Tillage requires big plots and a lot of space between plots. So what we could do is to think about, we could make bigger plots for tillage and then split each plot into parts and randomize with control methods to these smaller parts. So what we do is a design like this. You see, we have couples of small plots. So this would be a main plot, big one, and it would be in total 16 meters long, 8 and 8. And this big plot, and the tillage methods are allocated to these big plots. So you see here we have SP for the whole main plot. Here we have DP for the whole main plot. And here again we have minimum. So we have one block here, and there are all 3 tillage methods in randomized order, right? So we have a big plot for tillage. Then we split the plot, the main plot, in 2 parts. These are called subplots, and we randomize the 2 with control methods to subplots. Here you see we have part and tot. Again, here we have thought and parts. And here again, parts and thoughts. So you see, we have another constraint. The first constraint is that main plots needs to be organized in blocks. And this is the first constraint to randomization. The second constraint is that each main plot must contain all the levels of the second factor. This is called split plot design. It is common in medicine as well. A few years ago, I worked with some medical doctors from Sicily. They were, for some reasons I will not tell you, they were studying the diameter of the arm of ladies after an operation. And they were using the lady as a subject, as the main plot, and the two arms as subplots. And that was a split plot design. Well, it might look funny because we are not splitting ladies, but it is just figurative. I mean, we have a lady with two measures taken at the different arms. I mean, we could take one lady for the right arm and another lady for the left arm, for measuring the left arm. But that would not be rational. So we can use both arms in one lady. And this is a split plot. And it is the same if you want to make measurements of the eyes and if you want to study the difference between the right eye and the left eye. You can use different subjects for the right eye and the left eye, but that's not rational. You'd better use the same subject and you split it in two parts, the right eye and the left eye. So that would be a split plot design. So you see that the split plot design is not at all peculiar for field experiments. Right? So if you're clear with this, I will just show you briefly another type of design like this one, which is very common in the field, but it is also common in the laboratory. I've been working with this design several times in the laboratory, and there are also examples in literature. This would be the strip plot design. In this case, we make treatments in strips, which is handy. Look at this one. We have two blocks here. One is the left block, and the other one is the right block. This is the right block, and this is the left block. Let's consider the right block. This is a factorial where we want to compare 3 crops, rape, sunflower, and sugar beet, and 2 herbicides. One is rimsulfuron, and the other one is the control. So no treatments, no herbicide. Instead of considering the small plots here and treat them independently, what we should do, we do a different things. It is very handy to sew along the row here so that we can sew 2 plots at the same time and spray the herbicide along the columns so that we can spray 3 plots at the time. This is very handy because we have all the combinations. Here we have rape with rimsulfuron. Here we have sunflower with rimsulfuron, and we have all the 6 combinations. But we need to treat less plots because we treat them in groups. And this is okay because we do that in a randomized order. But we have put a further constraint. We have put a constraint to run to full randomization. We put a constraint for the block. We put a constraint for the rows. The rows must contain all crops. And there is a further constraint for the columns, which must contain all treatments. It's something like it reminds a little bit of the Latin square design, but this is different because we have two factors. With Latin square design, we have only one factor. And here we have two blocks, two separate blocks. This is called strip block design. In some other instances, you find it named as a split block design. But I think the most correct name is strip block design because we treat the plots in strips. And we can use this kind of design also for humans and animals in several instances. In laboratory, it's very common when we have to treat these small dishes with a lot of holes, and we can organize them in rows and columns. And then we use a strip plot design. What is important about these designs is that when we make data analysis, we should never forget that we have put constraints to randomization. And this is the problem with people working in the laboratory. They use constraints at the design stage, but they do not account for those constraints when they make data analysis, which is terribly wrong. It makes the analysis invalid. And if the reviewer realizes this problem, it will reject the paper. So we need to be careful to these types of design. We need to be able to recognize them, and then we need to be able to analyze them properly. But this is another thing which we talk about in the future. Just a brief mention of a couple of different designs. The first one, a subsampling designs. So we said that if we take one sample of sunflower seeds and we make three different analyses, for example, about content in oil. So if we take one sample and we make 4 analyses, these are not true replicates because these sunflower seeds are taken from the same plot, so they are not independent. These are pseudo-replicates or sub-replicates. But what we can do, and what we do very often, is to make a design like this. You might recognize this design. What is this design? Good, very good, Ugo. It's a Latin square. Good, very good. Oh, I'm happy because that means you're following me, at least 3 of you. You see that each treatment is in all columns and all rows. So B, 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 and B, C, 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 and C, and so on. So this is a Latin square design. So this is a good design because we have true replicates. A is in plot 13, in plot 2, in plot 11, in plot A, and these are independently treated. So we have 4 true replicates. Then from each plot, we take the sunflower seeds and make 4 analyses. So we have 4 true replicates and 4 sub-replicates per each replicate. In total, we have 16 values, 16 measures for each treatment. But we need to be very strict in keeping true replicates and sub-replicates parted. So we do not have 16 true replicates. If we say so, we are pretending to be more precise than we actually are. And that would be a mistake. We need to keep true replicates and sub-replicates parted during the process of data analysis. So this would be a subsampling design, which is correct as long as you keep true replicates and pseudo-replicates, sub-replicates parted during data analysis, right? And here we have another example, which is very important. So this is a factorial experiment. Well, indeed, this is a genotype experiment. So this would be a completely randomized complete blocks design. You see, the colors are the genotypes, and the yellow one is for the genotype A, the green one for the genotype B, and so on. So here we have one genotype, here we have one genotype, here we have the same genotype, and here we have the same genotype. What type of design is this? Do not consider the splitting, just consider the genotype. So again, the same question. What type of design is in the figure? Considering only the genotype factor. Someone said it is a randomized block design. Well, perhaps that was my original intention, but then I made a mistake, because you see, we have all 4 genotypes in the first block here, but in the second block, the yellow genotype is missing, and in the third block, the green genotype is missing, while we have 2 yellow genotypes. So by looking at this picture, the experiment is a completely randomized one. However, perhaps this is just a mistake on my side, because I made the wrong coloring, so I will correct it. The way it is now, it is a completely randomized design. But you see, I've written that there is the block here, so there must be a mistake, which I will correct immediately after this lesson. So this is a completely randomized design at the moment. And this is a perennial crop in particular. This was alfalfa, which is looser, which may be called. And you might know that alfalfa is a crop which lasts for 3-4 years in the field. Let's say 4 years. So I take this plot, the yellow one, bottom left, and I harvest it 4 times in 2001, 2002, 2003, and 2004. So you might recognize that this is a split plot design, because we have 4 splitted plots, although the splitting is not on a spatial scale, it is on a temporal scale. So here I have transformed a temporal succession in a spatial succession, just to show you. But this is a split plot. We call this design a split plot in time. It is very important, not only in the field. It is important every time we repeat measurements time after time. And we do that very often with animals, with humans. This can be regarded as split plot in time. More generally, these kind of experiments are called repeated measures experiments because we repeat measures in the same subject. And in this case, the subject acts as a block, as a constraint, as a so-called blocking factor. So I think this is all for experimental designs. What I want to tell you at the end is that the perfect design according to Fisher's recommendations is the completely randomized design. Because in that case, we put into practice Fisher's recommendation in the most strict way, because we randomize everything. But we have seen that complete randomization might not be useful, might not be handy, might not be precise enough. Therefore, we are allowed to put constraints to randomization, but we need to respect, again, Fisher's recommendation. That is, analyze them, meaning the experiments, analyze them as you have randomized them. So if you have used a particular type of randomization, then you have to analyze the data by using that type of randomization. So you cannot design an experiment in blocks and then analyze it as if it were a completely randomized design. That would be a mistake. That would make the experiment invalid. That would lead to the rejection of your paper. And that's all for the design.